Hello everyone, and welcome to our channel, MooTechie. It's so great to have you here. Today, we're embarking on a visual journey through the fascinating world of biochemistry. We're going to explore the incredible machinery of life, enzymes, how they're classified, and the helpers that make them work. Get ready to see how these tiny molecules power every process in your body. Let's dive in. Now let's move on to our first visual. Next, we explore what enzymes truly are. Enzymes, fast, specific catalysts. They are biological catalysts, which means they lower the activation energy needed for reactions to happen. They do this with incredible specificity at their active site, forming what's called an ES, or enzyme substrate complex. They are officially classified by the reaction type using the IUBMB EC system. And a crucial point, an apoenzyme, which is a protein part, plus a cofactor equals a holoenzyme, which is the active form. The diagram here shows a mechanism. A substrate enters the active site of an enzyme. They form an enzyme substrate complex. The reaction happens, products are released, and the unchanged enzyme is ready for another round. Coming up, we'll discuss the master system for organizing all these enzymes. IUBMB enzyme classification, EC1 through 6. We have EC1, oxidoreductases, EC2, transferases, EC3, hydrolases, EC4, lyases. Remember, an EC number is structured as class, subclass, subsubclass, and a serial number. There are 6 total classes from oxidoreductases to ligases. A handy memory aid is LILHOT, which stands for ligases, isomerases, lyases, hydrolases, oxidoreductases, and transferases. The visual here is a helpful hexagonal map connecting all 6 enzyme classes, giving you a clear overview of their relationships. Following this, let's understand each class in detail, starting with the first. Class 1, oxidoreductases EC1. These enzymes catalyze redox reactions, which involve the transfer of electrons between molecules. They often use coenzymes like NAD+/NADH and FAD/FADH2. Common examples include dehydrogenases, oxidases, and reductases. Their role is absolutely central in metabolism and cellular respiration. The slide has a bold, clean graphic with the word oxidoreductases. As we continue, let's see what gets transferred in the next class. Class 2, transferases. These enzymes transfer functional groups from one molecule to another, groups like amino, methyl, or phosphate. Think of kinases, transaminases, and methyltransferases. They play a central role in cellular signaling and biosynthesis. Their operation depends on a specific group donor and a specific acceptor. Now let's move on to a class that uses a molecule we all know very well. Class 3, hydrolases. These enzymes cleave chemical bonds using water, a process called hydrolysis. You interact with these daily. Examples are proteases that break down proteins, lipases for fats, nucleases for nucleic acids, and amylases for starch. They have a key role in digestion and the general turnover of molecules in the cell. And they're also widely used in biotechnology and various industries. The slide features a simple, effective graphic of a water molecule, H2O, next to the word hydrolysis. Moving forward, we'll look at a class that works without water or redox. Class 4, lyases, EC4. These enzymes add or remove groups from substrates to form double bonds. Importantly, they do this without hydrolysis or redox reactions. Examples include decarboxylases, dehydrotases, and aldolases. They often cleave carbon to carbon, carbon to oxygen, or carbon to sulfur bonds. The slide presents the word lyases in a stylized format. Next up, a class of molecular rearrangers. Class 5, isomerases, EC5. These enzymes catalyze intramolecular rearrangements. They change a molecule into its isomer, same chemical formula, but a different structure. This involves changes in stereochemistry or the positional shifts of functional groups within the molecule. Examples are mutases, epimerases, and racemases. Their role is crucial for balancing and interconverting metabolic intermediates. The slide has the title Isomerases prominently displayed. Let us now look at the builders, the final class. Class 6, ligases, EC6. These are the molecular joiners. They connect two molecules together, and this process requires an energy input, typically from ATP. They form important bonds like carbon-carbon, carbon-nitrogen, carbon-oxygen, and carbon-sulfur. Examples include synthetases, carboxylases, and the very famous DNA ligase. The role is essential for anabolism and biosynthesis. They build the complex molecules of life. The slide includes a symbolic representation of an ATP molecule. This brings us to a critical concept. Enzymes rarely work alone. Cofactors, enzyme helpers. This slide uses a simple, powerful visual flow. We start with an inactive apoenzyme. It meets a helper cofactor. Together they form an active holoenzyme. So a cofactor is a non-protein helper, which can be a metal ion or an organic compound. Remember the equation. Apoenzyme, which is inactive, plus a cofactor equals a holoenzyme, which is the active form. Types include metal ions like zinc 2+ and magnesium 2+, coenzymes, and prosthetic groups. Their binding can be loose like a co-substrate or very tight.
So what's the difference between some of these terms? Let's clarify. Coenzymes versus cofactors. First, cofactors. This is the umbrella term for all non-protein helpers. It includes metal ions like zinc 2+, magnesium 2+, iron 2+, and also organic molecules. They can be loosely or tightly bound to the enzyme. This is primarily a structural property that defines their binding character. Now, coenzymes, these are a specific type. They are organic cofactors often derived from vitamins. Examples are NAD+, FAD, coenzyme A, and TPP. They act as mobile carriers transferring chemical groups between multiple different enzymes. This is more of a functional property that defines their catalytic role. Among these helpers, some form incredibly strong partnerships. Prosthetic groups, tight partners. Let's start with the definition. A prosthetic group is a tightly bound cofactor, sometimes even covalently attached, that is essential for an enzyme's catalytic or structural function. Here are the key characteristics. They are tightly bound, often covalently, and essential for activity. They play catalytic or structural roles, like in glycoproteins or lipoproteins. An apoprotein plus a prosthetic group equals a holoprotein. Importantly, they cannot be removed without denaturing or unraveling the protein itself. Now for some common examples. Heme, an iron-containing group found in hemoglobin and cytochromes. Biotin, covalently bound, it acts as a CO2 carrier in carboxylases. PLP, pyridoxal phosphate, vital in transaminases, and TPP, thiamine pyrophosphate, involved in 2 carbon transfers. Let's zoom in on some of the most famous coenzymes you'll encounter. Key coenzymes, NAD, FAD, and CoA. First, NAD+ and NADH, nicotinamide adenine dinucleotide. This is a primary electron carrier for dehydrogenases. It's central to redox reactions in metabolism. It's loosely bound and mobile between enzymes, and it's key in cellular respiration. The slide shows its molecular structure. Next, FAD and FADH2, flavin adenine dinucleotide. This is a flavoprotein cofactor. It also drives redox reactions and electron transfer, but it's often tightly bound, acting as a prosthetic group. It has a major role in processes like fatty acid oxidation. Its structure is shown here. Finally, coenzyme A. This molecule is an acyl group carrier using its thiol group, that's SH. It famously forms acetyl-CoA, a central molecule in metabolism. It's pivotal in the Krebs cycle and is also key for fatty acid synthesis. Its structure is displayed. Now let's revisit and reinforce the concept of those inseparable partners. This slide repeats the detailed information on prosthetic groups, type partners, covering the same definition, key characteristics, and common examples like heme, biotin, PLP, and TPP, as explained previously. All right, we've covered a lot of ground. Let's bring it all together. Summary, what to remember. First, the EC system, 1 through 6, defines reaction types: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Remember that enzymes speed up reactions by lowering activation energy, and they aren't consumed in the process. The transformation, apoenzyme, which is inactive, plus a cofactor, gives you the active holoenzyme. And remember the distinction, coenzymes are organic and mobile, while prosthetic groups are tightly bound. Our key coenzymes are NAD+, NADH as an electron carrier, FAD, FADH2 in phleboproteins, and CoA for acyl transfer. And that brings us to the end of our journey. Thank you for watching this presentation. Subscribe to MooTechie for more educational content. Stay curious, keep learning, like, subscribe, share. MooTechie.